everyone, and welcome to today's webinar, 19th one in the series being organized by Manav Human Atlas Initiative. My name is Jyotsna, and I'll be the moderator for today's session. For today's webinar, we have Professor Gautam Menon, who will be speaking on infectious diseases and how to think about them. Dr. Gautam Menon is a professor of physics and biology at Ashoka University, an adjunct professor at the TIFR Mumbai, and professor at the Institute of Mathematical Sciences, Chennai. He has spent 2 decades as professor with the theoretical physics and computational biology groups at the Institute of Mathematical Sciences, Chennai, where he was the founding dean of Computational Biology Group. He is a physicist and mathematical modeler by training, with interests in biophysics, disease epidemiology, and science communication. He works in mathematical modeling in a range of fields, including the modeling of COVID-19. For today's talk, Professor Menon will describe models for the spread of COVID-19 in India and discuss results from these. He'll also illustrate some of the difficulties that modeling presents and the need for better data to constrain models. He'll also be showing us how modeling can help frame better health policy and why this is especially crucial for Indian scenario. Over to you, Professor Menon.
Thank you, Jyotsna. It's an honor to be in a series. I want to tell you a little bit about the work that we've been doing over the past year, more than a year, on models for COVID-19, but I want to set that in the general context of thinking about diseases. And in order to do that, I'll have to tell you a little bit about what infectious diseases are, so that we understand very clearly what should go into the modeling of infectious diseases and what the consequences of that modeling might be. So infectious diseases, as we've known for the past approximately 150 to 100 years, are caused by a microorganism. It could be a bacterium, it could be a virus, it could be a parasite, it could be a fungus. But these are all sort of small, living, often unicellular organisms. And some of them are even straddle the boundary between living and non-living, such as viruses. If you look at infectious diseases in their categorization, H1N1 is a viral disease, dengue is a viral disease, malaria is a parasitic disease caused by the malaria parasite, influenza is a viral disease, the flu, tuberculosis is a bacterial disease, MERS is viral, SARS is viral, and COVID-19, the pandemic that we're living through now, is also a viral disease. Bacteria are living organisms. They multiply rapidly inside your body. It's your body's immune response to the bacterial infection that makes you feel sick. And antibiotics attack bacteria in multiple ways. They can break down the ways in which bacteria reproduce. They can break down the cell wall of bacteria. And we know increasingly more about the actions of specific antibiotics on this. And of course, the worry is that as bacteria develop resistance to antibiotics, we have to search more and more for ways to subvert that resistance and make sure that our ability to treat diseases that are bacterial remains intact. Viruses sit at the boundary of living and non-living. They multiply inside cells and then escape, often by lysing the cell, by breaking it up, in order to infect other cells. Antibiotics, of course, don't work against viruses. They're not constructed to work against viruses. And the treatment of viral diseases is a little more complicated than our understanding of the treatment of bacterial diseases. Why don't we always constantly fall ill? And the reason is that we have an extremely well-developed and remarkable immune system that protects us. Any prior contact with the disease, with a bacterium or a virus, helps the immune system recognize the invader. And when you vaccinate, when you immunize, what you're doing is really priming the immune system so that it recognizes it when it actually comes around. You're priming it with little pieces of whatever it is that might infect you later, so that your body is able to mount a suitable immune response when it actually comes, when it's actually challenged. Bacteria and viruses move between humans directly or indirectly. A direct contact between people is one way of moving a virus from one person to the other, from a person who's infected to a person who's susceptible. But there's, of course, also indirect contact. If someone who's ill touches, sort of wipes their face and then touches a doorknob, and someone else comes and touches it later, that's a way of transferring bacteria and viruses from a person to another person. Droplets, something that's really important in the context of COVID-19 is one way where you sneeze, you emit small little droplets that contain phlegm, that contain little pieces of virus, and the virus is ingested by someone else who happens to be standing by. And in some cases, for airborne viruses, it can just stay in the air for a while until it's ingested at some later point. Some diseases come to us from animals, for example, from bats, for example, and poultry, as is currently is a problem. These are called natural hosts for their viruses. And an event in which a virus moves from a natural host, which is a bat, poultry, an animal, to a human being is called a spillover event. SARS, MERS, and COVID-19 are all examples of spillover events where you had viruses that technically belonged and were perfectly happy in animals, but somehow made the transition to coming and infecting human beings. This is interesting because, as we know now, among the new and emerging diseases, 3 out of 4 of them typically come as spillover events from animals. So it's increasingly important to understand the sources of such spillover events and to ensure that this is minimized as we go along. And these intersect many questions of ecology, of human-environment interaction, all of which combine into how we should think about diseases of this sort. Diseases are old. There's nothing new about it. Smallpox was described in old Egyptian and Chinese writings from a few hundreds and thousands of years ago, and may have been responsible for more deaths from all infectious diseases combined. And there's evidence that malaria and polio have existed since ancient times, from ancient writings. Diseases have been responsible for the collapse of entire populations. The Han Empire in China in 3 AD was presumably destroyed by plague. The Aztecs were taken over by the Spaniards, who spread smallpox among them. This is a population that was naive and had never seen smallpox before and had no defenses against it. Plague killed 3 Europeans out of 10 between about 1347 and 1351 over a fairly short period of time. And the biggest pandemic of our time before COVID-19 was actually the 1918-1919 pandemic flu, in which somewhere between 60 to 100 million people died. So let's just go into that a little bit, because as I said, this is probably the defining pandemic of the last 100 years. And it's possible that COVID-19 will also become a similar defining event of public health across the last 100 to 150 years. This is a picture of a ward of people together, all of them affected by influenza, by the Spanish flu, that really spread through the world. So it was a true pandemic in the sense of affecting many, many more countries other than the country of its origin between January 1918 and December 1919. The reason so many people died, and there was a large number, it infected around 500 million people altogether. That's about 25% of the world population, killing somewhere between 30 to 50 million. We don't have a very good handle on that number. It's all very approximate. But one interesting thing is that it hit India probably harder than it hit any other country. And somewhere between 50 to 20 million people died in India alone. Many of the main figures in our freedom struggle, including Gandhiji, were affected by the epidemic of Spanish flu. And at that time, this was 5 to 10% of our whole population. So that's what pandemics and epidemics in general can do. You have people who are seriously affected by it. They may carry the seeds of this long, even if they survive, and people will also die from this. And of course, the Spanish flu came at the end of a very bruising world war. This was World War I, where people had already been suffering through many days and years of bad diet, of bad nutrition, etc., and were infected, for example, by the rigors of trench warfare. So it probably spread faster than it would have done in a situation where the health of people overall was better, and the nutrition was overall better. So here's the timeline of the diseases that have struck us since the beginning of the century. So SARS was, of course, the pandemic of 2002, 2003. Didn't kill too many people finally, although it was a fairly fatal disease, but it was managed to be contained. H1N1 in 2009 infected a very large number of people across the world, but was not fatal except in a relatively small number of cases. MERS, again a fatal disease, killing between 30 and 40% of the people it infected, but stays in the background and doesn't really spread out throughout the world, although that is a serious issue in many parts of the Middle East, including Saudi Arabia, as well as other places where it has cropped up from time to time, which is South Korea, for example. Ebola, multiple sort of points at which it has written and then come down, including as recently as last year and parts of this year, and of course COVID-19. So this is just to show you that there is a continuum of disease. It's not as though there is any time where humankind is free of disease altogether. And each of these are, in a sense, unique in their own ways. Every pandemic is unique. Every epidemic is somewhat unique in the way it chooses to spread and where the geographic extent through which it spreads. So let's talk a little bit about COVID-19, because much of the modeling that I will tell you about relates to COVID-19. So COVID-19 is caused by a type of virus called the coronavirus, and because of its very characteristic crown-like shape. And the virus is called the SARS-CoV-2 virus because of its relationship to the earlier SARS virus. As far as we know, this originated in China, in the province of Wuhan. It spread worldwide. And now there are very few countries, probably none at all, that have not had at least one case of COVID-19. This is important that you know that it's not a treatable disease. There is no antiviral that works conclusively against COVID-19. All you can do, all a medical professional can do, is to provide supportive treatment. This spreads through a respiratory route, as I said, through droplets that move in the air as someone infected coughs them out. And that's why it's particularly good, particularly easy for it to spread across multiple people. And this is why also mask wearing is a good idea. Most people will recover from COVID-19, and some will actually not even know that they were ill. And this is interesting because this is an aspect of an infectious disease that is not completely common. The fact that many carriers of the disease will remain asymptomatic. They will not show symptoms at all, or they will show symptoms fairly briefly. We know now that you tend to see more adverse outcomes of the disease in older people. So being young protects you to some extent against the disease. And pre-existing conditions are important. For example, if you're diabetic, if you're overweight, if you have COPD, if you have other types of conditions, various types of immune conditions, you tend to be more prone to an adverse effect following infection with COVID-19. This is what the world situation looks like. And each of these circles is a circle for Europe, for Latin America, for Asia, for the Middle East, etc. And you can see that Europe has had the largest number of cases overall. And this is, you can see that this is counting the number of deaths in this case. So you have Britain, Italy, all with a large number of deaths, 87,000, 81,000. The US, of course, stands out with 392,139 deaths. India, a smaller circle, 150,000 deaths over there. And you can see that much of Asia, even though Asia contains much of the world's population, is actually a smaller circle. The size of the circle here roughly corresponds to the number of deaths. So the largest is certainly Europe with about 600,000 deaths, the US following next, South America again with a very large number of 545, but Asia, 545,000, but Asia is still smaller than that. And that raises a mystery. Is there something that protects the South Asian population vis-a-vis the rest of the world? Or is this just by chance? This is what the case in India looks like. And this is the number of daily positive cases since the pandemic started. So the numbers start off taking off by the 22nd of March. They peak around the 10th to 15th of September. And then after that, there's been a steady decline, barring a tiny little sort of small little peak somewhere around November. So this is extremely interesting. Unlike other countries such as the US, which have seen multiple peaks, it looks as though in India there has been really one substantial peak, and there's nothing to indicate a sort of further peak, certainly for the last 3 to 4 months. And it's interesting to ask whether that will continue or whether this is just purely by just a temporary stalling in the progress of the disease, and you would expect this number to come back up again. Let's zoom in a little more and let's look at what the state is across multiple countries. So this is the US, India, Brazil, Russia, UK, ordered in terms of the total number of cases, total number of people who've been infected. What I want to draw your attention to is what's in the last column, which is the deaths per 1 million population. The US stands out at 1,221 deaths per million. Brazil stands out at 982. But in comparison, even though India has had a large number of cases, more than Brazil, the number of people dying per 1 million population is small. It's a factor of 10 smaller than the number for the US. And again, this is interesting. Are Indians protected in some way against sort of the worst manifestations of the disease, mortality? That's an interesting question. Could some of this be accounted for by the fact that we're not counting the deaths right? That's also a possibility. But is that enough to make up for this difference? We don't know. So all of this remains very interesting questions about the nature of the disease as it affects India. So as I said, let's zoom in a little more on different parts of India. So this is the cases, the total number of infected people, the daily count of cases in Delhi, Bangalore, Pune, Mumbai, Thane, etc., etc. So what I want you to notice here is that each of these curves looks very different, and they all look very different from the India curve. The India curve showed a single peak and then came down. But here in Delhi, you can see at least 3 peaks. In Pune, you can see a smaller peak and then a second peak. Mumbai, you see a sort of little peak, but then never really coming down, and then a somewhat larger peak later, which is not the case, for example, in Thane, which shows a peak, a decrease, and then a subsequent peak. So what accounts for this diversity? Where does it come from? And is there any way of trying to understand where this variation might be? Can one predict some of this variation? Can one understand some of this variation as a consequence of various measures that governments, state governments, local governments took in order to deal with the disease? So here is the picture. I just took 4 examples from here, from Delhi, Bangalore, Pune, and Mumbai. So questions that we can ask at this point is, how do we understand this very different type of behavior? What might happen in the future? What would vaccinations do? Is vaccination a way of pushing that number all the way down to 0, or can we still expect that there might be more cases? Who should we vaccinate first? What is the order in which we should do it? And how do we use models in public health effectively is a larger question. As a modeler, as a person who thinks about mathematical models and how they might apply to real life, this is something that I'm very concerned with. Or what is a sensible way of thinking about models in such a way that you can provide information to people who make policy about public health that enables them to make better decisions. So I want to tell you about 3 pieces of work that I've been involved in. One is called IMSc. I will tell you a little bit about network models, and I'll finish with a piece of work that is still very much ongoing called Bharatsim. Okay. But before that, let me just tell you a little bit about the details of how we think about diseases in populations. What is the information that we need, and how do we encode that into mathematics and computational programs? When we talk about disease in a population of individuals, the first thing to remember is that every individual is different from every other individual. If you're healthy, if you have no immune problems, etc., you might be less susceptible to a disease than someone else who's immune compromised, 80 plus, suffers from a raft of other types of conditions at the same time. But it's often the easiest thing to do to simplify this and assume that everyone is either uniformly susceptible to the disease, they're already infected by the disease and therefore capable of infecting other people, or they may have recovered from the disease. So these little circles here represent individuals, and I have characterized them by saying they're either susceptible or S, they're infected or I, or they're recovered or R. So in this case, I have only susceptible and infected people in this population. The susceptible people are not exposed, not infected, but they could in principle become infected if they encounter someone who is infected. An infected person is also infectious. They can transmit the infection to susceptible people. And once you recover, let's assume that you don't catch the disease again. So you have recovered from the infection. One way to now think about it is to imagine that you put everybody in the population into an appropriate box. There's a susceptible box in which you put all the susceptible people. There's an infected box in which you put all the infected people. And there's a recovered box in which you put all the recovered people. These are called technically compartments in the mathematical language. So you have people in these different compartments. You can't belong to 2 compartments at the same time. And as the susceptible person becomes infected, they move to the infected compartment. As the infected person recovers, they move to the recovered compartment. Okay, so that's the S, I, and the R that enter the model. The numbers in each of these compartments is what one tries to model. The number of susceptible at a particular time, the number of infected at a particular time, the number of recovered at a particular time. And that's the fundamental quantity that we want to try to answer. We want to try and ask, will the number of infected ever be such that it exceeds the capacity of hospitals to deal with them? At what stage will the number of infected rise to a maximum? At what stage will it begin to come down? Having come down, is it possible that it might go up again? And what might lead to that? These are questions that one needs to ask. One can take this description in words that I outlined over here and convert it to a description in equations. And that's a set of equations that I've shown you on the right-hand side. These are details which don't really matter to understanding what's going on. The understanding really came from the earlier picture where I have compartments and I have people moving from compartment to compartment. And at any instant, the number of people in each compartment is what is crucial. If I take these equations that represent that, what I described in words, and solve these equations, I get something that looks like the picture on the left-hand side. I have the number of infected people rises steeply, and then it comes down with a long tail. Along with that, the number of susceptible people comes down and then flattens out, and the number of recovered people goes up. So at any given time, given the simplicity of the model, the number of susceptible people plus the number of infected people plus the number of recovered people must always be a constant, because we're assuming that no one is dying and no one is being born into this population. And all of this we can change and make more complicated models. But this is, in a sense, the simplest model that one can think of diseases. It's a very famous model. It is called the SIR model. And there are parameters that enter this model. And from these parameters, one can make a very specific number called the basic reproductive ratio, R0. R0 is very important. It's, in a sense, a very fundamental characterization of a disease at the point where it begins to spread. The question that R0 answers is, given that there is one sick person and everyone else is susceptible to the disease, there's one infected person, everyone else is susceptible, on average, how many people will they go on to infect? There are a couple of things here. One is, how efficient are you at being infective to someone else? How many people do you contact? What fraction of those people will you infect? And how long will you remain infective? What is that period? So all of this enters into the calculation of the reproductive ratio. For Ebola, that number is 2. For swine flu, that number is 2. For HIV, it's 4. For measles, it's 18. So measles is highly infectious. If there's one person with measles and everyone else has never had measles before, that person is likely to go on to infect as many as 18 people on average before they recover and become non-infectious. You can look at the reproduction number as it is for COVID-19. And right now, this is a number that is a little less than 1, and that is important. If it's greater than 1, on average, you're infecting more and more people every time. So, for example, if it were 2, one person infects 2 people, who infect 4 people, who infect 8 people. Each time this doubles as you go along. And this is an exponential increase in the total number of people who are infected. So it's important to see at any given time what is the value of the reproductive ratio, and to ensure all our interventions really go into pushing the reproductive ratio down below the value of 1. It's a little more complicated to talk about COVID-19. It's not a simple SIR model, because for 2 reasons. One is that you can be exposed to the infection as your virus builds up in your body, and then during that period, you're ill because your virus is multiplying in your body, so you're no longer technically susceptible, but you haven't reached the threshold at which you can be infectious to other people. So that's called an exposed compartment, which we must add to the S, I, and R compartments. The other thing to be note for COVID-19 is, as I said, many people will be infected with COVID-19, and yet they will not have any symptoms. So they will not know that they are ill, and they will not be in any discomfort otherwise that might lead themselves to quarantine themselves or to prevent themselves from meeting other people. So this is called a pre-symptomatic period. This is the period in which you can infect other people, but you don't show symptoms. And it's important to account for that for COVID-19. The first piece of work that I want to talk about is some work that began as a subgroup of the Indian Scientists' Response to COVID-19, or the ISRC, and you can look up its webpage. There are a bunch of reports that we wrote based on the model that we created for COVID-19, and all of these reports are available on the webpage. I want to summarize a few of those results for you and to tell you what they mean. So the group that we were working with, the Indian Scientists' Response to COVID-19, made a model called INSCISIM, for Indian Scientists Simulations of COVID-19. This is like the model that I told you earlier. It's a compartmental model. It's a compartmental model intended to represent COVID-19 in India. It's just a much more complicated model than the relatively simple SIR model that I showed you, which was the simplest of the compartmental models. One thing that we did was to try and benchmark rates for the modeling of COVID-19 in India. We wanted to include demographics, the different populations of different states, etc., as well as migrations, motion between these states. We wanted to look at the effects of various interventions. For example, what does a lockdown do? What does an increase in testing do? What does masking do? All of these we wanted to put into the calculation to try and see how does one change or how does one alter the total numbers of infected. There's an online tool that is available that you can all check out. The tool is designed to be easily updated and to make sure that all the changes that are recorded in the tool are made transparent. So this is what really goes into the model. And as I said, it's a little more complicated than the standard SIR model. It says that there is some category of people who, when infected, they are asymptomatic altogether and they will recover relatively fast. Maybe they're self-sick for about 5 or 6 days, but after that they recover. People who are mildly ill will show symptoms. For some amount, they will go through a pre-symptomatic state. They will show a mild symptoms infected state, and then they will recover. But there are also people who are more severely ill. Severely ill people will go to hospital and they will recover. critically ill will also go to hospital, may require ICU support, may require ventilator support, and some fraction of them will die. And this is the progress that they take through the different stages of the disease that is described here. The model parameters that go in are described. The equations are as below. You can put various, the sort of the picture in terms of compartments is the picture that you see below on the left-hand side. And you can generalize this in various ways. And all of this is things that we have done. But let me just take you through some of those generalizations, just to tell you the complexity that is required of a model. The first thing that you can do is that since you know that the disease affects people of different ages very differently, people who are young can be affected. They can carry the same amount of virus as people who are old, but somehow they don't seem to show symptoms as often or as consistently as older people who tend to be much more affected by the disease. So we can divide each compartment into different subcompartments that represent the 0 to 10 age group, the 10 to 20 age group, 20 to 30, all the way up to the 80 to 90 or 90 to 100 age groups. These are called age-structured compartments. The second thing that we can do is take social information, social survey information, and try and understand how often does someone who's 80-year-old contact physically someone who's between 20 and 30, as opposed to someone in their same age bracket. What do we know in India? And this is important because in India, we still have families who live together, multi-generation families who live together in the same house, where grandparents, parents, and children all interact together. And this is often, and the way societies are structured is different between, for example, the industrialized West versus India versus other parts of the Far East. And the nature of contacts between different age groups is in fact dependent upon these interactions. So these contact matrices enter the calculation that we do. We work with what is called metapopulation, and metapopulations defines, for example, different states, Chennai, Maharashtra, Tamil Nadu, Bihar, Andhra, etc., and describes each of them as a population in its own right, but then also accounts for the movement of population, for example, the migration of workers from Maharashtra back to UP or from UP to Delhi, etc. All of these are things that we can put into this description, this mathematical description of the model, and then query as we go along. The model itself is fairly complicated. As I said, there are 9 compartments, there are 8 age groups, there are 740 districts in India, roughly speaking. That's a lot of equations, and these are all equations that have to be solved simultaneously and compared to the data that is available through some nonlinear minimization. We don't do it at this level of complexity yet, but it's there in the model. So right now we can study individual states, individual cities, etc., without worrying about the problems of how do you account for migration from different parts and how those change the numbers. So right now we can look at districts in India. It's an age-structured model. The contacts are there. We can put in migration. We can put in healthcare accessibility. How easy is it for someone who is severely ill to go to a hospital versus not able, versus an inability to? And we can also look at the quality of healthcare that one can supply to these people as a function of each district. There's a dashboard that exists that is available at this particular website that you can actually run your own simulations, change your own numbers, and look at what might happen. Here are some tests that one can run on the model. For example, if you reduce the infectivity of the people, if you just make it that much more difficult for people to infect other people, for example, just by ensuring that everyone wears a mask. If everyone wears a mask, it's less likely that the viral particles inside your chest, your throat and chest will be spit out and infect other people when you go and encounter them. So that will be a reduction in your infectivity. And you can see that the numbers of infected reduce as you reduce that particular number, the number that means infectivity in our model. And also the number of hospitalized people will also reduce. So this is called flattening the peak, any intervention that helps to reduce the total number of people infected. You can also ask about what happens if you put in testing and quarantining, if you put in lockdowns, etc. And a testing and quarantining protocol imposed so that it slowly ramps up over a period of 100 or 200 days also is very successful in reducing the number of people who are infected. So various types of what are called non-pharmaceutical interventions, a lockdown being just one of them, can help to reduce the value of the peak. We could also ask what are different ways in which people can come back to work, and would something like a staggered schedule work? For example, you work for 5 days, then you're quarantined for 4 days, and you work for 5 days, you're quarantined for 4 days. And suppose this mix of N days and M days together. And of course, the moment you show symptoms of the disease, you're pulled out and isolated. Does this help in getting larger number of people back to school, back to colleges, back to work? We can explore all of these different varieties of scenarios within the model. Here's some work that we did in which we actually looked at Pune. from where Manav Atlas at least partly is being run. The little red dots that you can see are the number of cases, of critical active cases that required hospitalization and how they increased from April to May to June. And these were different scenarios. All the lines that you can see were different scenarios that we tried to plot out at that time for how this could increase. At this point, you can only see that it's increasing, but you don't know which route it might take. A little later, we did this more carefully and we defined certain widths in which we expected the behavior to be. And the dots there are again the regions in which those represent the actual number of hospitalization. And the band that you can see, the gray band is the band that we said, it's likely that this number will sit within that particular band. And this was our prediction for what it might be in the future and where it would go. And we sort of kept updating this as you went on in time. So this particular figure is from November 14th. By Jan 2021, you can fill in more data, and you can see that you left that, you went on the lower side of that band, and then you're back in that band. So the fact that it remained within this predicted band is in some sense interesting. It never really went to the worst case, but managed to be controlled. And we were able to sort of show that it would remain within this rough region using the modeling techniques. We've been doing this for many other parts of India. For example, for every district in Karnataka, we have numbers that are week-to-week predictions of what you might expect in the coming week, what might happen, where it's again compared to the observed data. So there are a bunch of predictions here. This is work done together with a group at the ISI Bangalore and a group at the ISC Bangalore. And you can see that how our line compares, along with other models they have tried, to the actual data. It sort of goes up, and you can see the saturation value, both, for example, Mysore here and Bangalore urban in the bottom right corner. If you look at how our model does, for example, for Mumbai, for the daily infected in Mumbai and the daily deaths in Mumbai, and the data is, of course, very noisy, jumps all the way up and down, but the rough shape of this, you can see is captured. The multiple peaks that happened in Mumbai is captured in this particular calculation. Delhi, for example, we looked at Delhi and then asked what would happen if you relaxed any intervention on the 1st of August. And you can see that the curve was initially coming down. So that's hospitalizations and infections. But we said if R was relaxed at that point, if people suddenly went back to work, back to contacting each other, public transport began to take over, then you might have expected an increase. And that increase is shown for different assumptions as to what the infection fatality rate might be in this case. And you can see that Delhi actually did see this increase, kind of what we suggested might happen if you relaxed the fairly rigorous constraint that had existed until about August 1st. And at the bottom right-hand corner, you can see the comparison between the projections and what the numbers actually look like. Then the second part of the talk, let me actually go to telling you a little bit about network models and finally wind up with Bharatsim. Network models, our work on network models was targeted at the following question. Right now, if you want to be tested for COVID-19, you can take one of two possible tests. One test is the regular RT-PCR test, which is an accurate test, but it takes a little bit of a delay. If you test it today, you get your results back tomorrow. The second type of test is called the rapid antigen test, or the RAT test, and that is a point-of-care test. You can take it now and get an answer within about 15 minutes. It's called a lateral flow assay test. Right now, India tests about less than 0.1% of its population on a daily basis. Contact tracing, that is, if you are infected, attempt to identify all the people who came into contact with you and get them tested, that's not really happening across much of India. Most of the testing happens when someone reports with symptoms to a particular COVID testing center and says, can I get tested? And of course, the other scenario is when you go to hospital for regular admission for something else, and then they will test you for COVID in order to ensure that you don't infect anyone else. The current procedure is to isolate a person who tests positive for COVID to ensure that they don't infect anyone else. The ratio of how many tests are RAT versus how many tests are PCR is controlled by states. And this ratio has tended to be somewhat skewed in some states. For example, across India, the ratio is somewhere between 40% to 60% on average. But individual states can do much more RAT testing versus PCR testing. For example, Bihar, Delhi, and UP do less than 50% of PCR tests. The reason this is important is that the accuracy of these tests are not the same. The RAT is a medium accuracy test. It's not very accurate. If it's about 50 to 60% of the time, it will correctly say that you have the disease when you do have the disease, but some amount of time it will tell you that you don't have the disease, where the test goes wrong. On the other hand, the RAT is an inexpensive test. So governments like these tests because they can roll out a large number of them. for a fraction of the cost that it would do to do a PCR test. The RT-PCR test is a high accuracy test. Its sensitivity is 98% or 99% in the laboratory. But on the other hand, as I said, it takes a delay. And often the problem with testing is there are delays between the time that you are tested and the time that you get your results back. If you're lucky, it can take a day, but there have been times where it can take 5 days to 7 days for those results to come back. So then the question is, how much should you be testing? What is a mix of RAT to PCR that you must be testing at? What is the sensitivity of these tests that you must aspire to? And who should you choose or who should you prioritize for testing? So these are all questions that need models to think about. And the model that we used is what is called a network model. So the network model has people who are at home. So people are assigned to homes across with a certain distribution of family sizes. The mean family size is about 4, but you can have families with 5, 6, 7 people, as well as families with 1 and 2 people in a particular, for example, in this model. People move from home to workplaces. So workplaces have typically somewhere like 20 to 40 people in them. So people go to workplaces, they come back, and sometimes if one of them is infected, they can infect both the people at home as well as people in their workplaces. The idea is that the moment someone begins to show symptoms of the disease, they can get tested for the disease. They can be tested either by an RAT test or by a PCR test. And you can vary the fraction of people who are tested by RAT or by PCR. You can also vary the amount of testing. Given a population of size 100,000, you can say that, look, I will test 0.5% or 1% or 2% of this population every day, because that is the capacity that I have for testing. People who are seriously ill can go to hospital, and then in hospital they can infect healthcare workers. So that's another question. Should we do anything special about testing healthcare workers, given that they're surrounded by people who are more likely to be infectious with COVID-19 than people they would intersect otherwise in the general population? So that's what these networks look like. You can see the pictures of the networks at an early time, and there are few people at an intermediate time where many people are infected, and at a final time when people have begun to recover. What we can do with this is the following. Suppose we say that a particular government, let it be the government of UP or Maharashtra or Bihar or Tamil Nadu, can test at a certain fraction of their population, a daily test rate, which is somewhere between 0.1% and let's just say 0.8%. Right now, as I said, India overall tests at less than 0.1% of its population on a daily basis. And the states don't deviate very, very far away from that particular number. They sort of span this range that we can see. Given that you're testing at a certain daily test rate, suppose I have a test whose sensitivity I can vary all the way from 50% to 90 to 100%. And I can also vary the fraction of the RAT test versus the PCR test. Fraction of people will finally be infected at the end of the epidemic. If you didn't do anything at all, you did no testing, no intervention, anything, somewhere within this model, somewhere between 65 to 70% of people would be infected by the time the epidemic begins to come down. But now you can see, can I push that number down by testing, for example, at 5% of the 0.5% of the population, by testing with a test that has a sensitivity of about 60 to 70%, and with a mixture of RAT and PCR, that is 60% RAT and 40% PCR. So that will tell you exactly where in this grid the sort of value that you might reach is. And you can see here that as you increase the fraction of RATs, you tend to have more and more people getting infected. So that's a rise as you increase the fraction of RATs. And as you increase the sensitivity, you tend to come down. The more sensitive you are, the more cases you will begin to pick up. And therefore, you will quarantine these cases and prevent them from infecting other people. So all of this information you can get from curves that look like this. And the other thing that you can do, of course, is to say, all right, let me add an economic component to this. And let me say that the cost of the PCR to the RAT is a factor of 2. PCRs are twice the cost of the RATs, or they're 3 times the cost of the RATs, or they're 1.5 times the cost of the RATs. Suppose I said, look, instead of 65 to 70% of people getting infected, I want to push that down to 50% of the people getting infected. What is the cost of this? If a PCR to RAT cost is 2, the cost fraction is 2, how can I change the sensitivity in the RAT fraction and see what the cost might be of a particular intervention? Obviously, as I increase the RAT fraction, I will find that it's cheaper and cheaper to do that because RATs are cheaper than PCRs. But on the other hand, more people will get infected because the RAT is less effective. So there's a trade-off between multiple things: the cost of the test, the sensitivity of the test, and the fraction of people that I can test. And all of this can be quantified in this way of thinking about it. So here's where models are important. You can see there are multiple trade-offs. How sensitive is your test? What is the fraction of the population that you can test on a daily basis? What is the mix of tests, the RAT to PCR, and how much do they cost? And what is the stage of the epidemic when your testing begins? Are you testing at the beginning of the epidemic, or are you testing halfway through the epidemic when 20% of people or 30% of the people have already been affected? There's no way of really answering these questions without a mathematical model or a computational model of the sort that I told you about. And that's where the nature of models and thinking about diseases from a computational and data science perspective becomes very important. The rough result, the later you test in the epidemic, you need to test more to achieve the same effect. You should target symptomatic people, and even a fairly large fraction of RATs is okay. The other interesting thing is that this synergizes with many other types of intervention. For example, the more mask wearing you have, the more you can reduce the amount that you can test, the amount that you need to test to achieve a specific reduction. And the synergies between multiple interventions is a very important nonlinear effect that leads to each of these interventions individually becoming more important than if they were practiced completely on their own. The last thing that I want to mention is Bharatsim, which is a large-scale project that we've been working on for the past several months. And in a sense, it's the most complex of all of these projects. Bharatsim is an agent-based model. That is to say, it tries to simulate every individual on their own. So agent-based models are models where you can imagine a city of Pune or a city of Bombay with somewhere between 6 million and 12 million or 13 million people, and try to put each of them on a computer and try to replicate the sort of behaviors that they might. People are at home, people are at work, they use public transport to go between home and work. If some of them are infected, they tend to infect people they come into close contact with. They may infect them in the transport, in the particular transport that they're using. They may infect them at work, they may infect them at home. You can have people who are sent to hospital where they may infect healthcare workers. All of this you can put in, but you can put in many, many other forms of detail when you deal with people at this very individual granular level. You can put in geographical detail. And as I said, Bharatsim is this ultra-large simulation of, in quotes, agents. It's an agent-based model. You derive the properties of these agents from what is called a synthetic population. And this is work that is done together with a company based in Pune called ThoughtWorks. There are three parts to this. One is, of course, a computational model to simulate these agents, how they move from place to place, how they infect other people, and what are the properties that affect that. If someone belongs to a particular socioeconomic class, is within a certain age group, what is the universe of people that they're most likely to infect? And all of this has to be put into the model. How agents interact, what is the interaction at school, what is the interaction at home, what is the interaction at work. All of this has to be specified using appropriate networks. And that's another part of thinking about this model from a computational perspective. And the last, and in a sense, a very important part of this is what is called a synthetic population for agents. And the synthetic population, the idea is to create a synthetic population for India that integrates multiple data sources. For example, for each city, for each state, for each district of India, what is the group, what is the division by gender, what is the distribution by age, what are typical family sizes and distribution. Can you integrate equal access data, education levels, immunity of into the description of the synthetic population. You can think of this as a very big file where each line represents one model synthetic person. And then you can use this in this model, but you can also use it for many other types of techniques and tricks that you might want to try out for public health questions. Here, this is being combined currently with machine learning methods that try to create realistic looking synthetic populations. And that is a challenge. And as I said, this is ongoing work. And the scale of this is very large. And this is a frontier in many ways that we want to think about. So let me summarize, since I'm coming to the end of my time. I've told you a little bit about infectious diseases, given you a background to what diseases, what have been the diseases in our past, what are the basic facts that you must know about disease. Then we talked a little bit about the history of COVID-19, where it started, how it spread, the fact that it really is a pandemic, and a little bit about important information about COVID-19 that any model must sustain. Then we spoke about epidemiological models. We talked about the SIR model. I talked about more advanced models than the SIR. And I said that COVID-19 required that you to accommodate a certain level of detail in the model description. Then I told you about three models for India. The first model was INSISM, which is a very classic epidemiological compartmental model. The second was a network model that we wrote down to answer a specific question. What is the nature of testing, and how is that affected by the fact that you have two tests that have different levels of sensitivity and that cost different amounts? What can we say about this? And the final thing that I told you about is very much a work in progress. That is the Bharat Sim program, which is an attempt to create a synthetic population for India, as well as a way of defining agents in a computational model that would derive from the properties of the synthetic population that we could use to study the spread of COVID-19. The last, Bharatsim really emphasizes the importance of data. You need to have good data to be able to create a synthetic population. That, in a sense, will be a hurdle for anyone who wants to try to do something similar. How does one integrate data across multiple databases taken at different times that are fragmented in various ways to create something that is realistic and can really represent a population that you want to study? But more than that, you also need models. You need to be able to work with data and try to push that data in surprising new directions. And that's where both modeling, data, computers, big data, et cetera, all of this come and play in together into addressing questions of public health, which are probably the most important questions that face us as a nation at this point. The next step would be to combine all of these models, the insights model, the network model, Bharatsim, into trying to understand what might be good vaccination strategies and vaccination protocols going forward. As you know, India started its large-scale vaccination program on the 16th of January. And the questions going forward for the next few months will be, what is the best way to target these vaccines, to use up the vaccines? What is the best schedule that we have for them? And as with the tests, we do have two different vaccines at the moment, both of differing efficacy. And we want to be able to try and understand what might the effects of that be on the population and on controlling disease within the population. These are all very interesting and exciting questions. They happen to be very current questions because they're all playing out in the background of the global pandemic, which has also hit India. And it's in whatever way we as computational scientists, as data scientists, as public health experts, we can work towards ameliorating the impact of COVID-19 on India. This is something that we should do. As always, any work of this magnitude has to be done with lots and lots of remarkable people, including a whole bunch from Pune. So there is a large group that has worked on the insights model that was worked together with the Pune Knowledge Cluster. Work on networks, testing and vaccinations has been with Sandeep Krishna, Philip Cherian, now with Brian Wall at Johns Hopkins. Devayan Gupta has been a long-term collaborator on the Bharatsim project, looking at the synthetic population. There is a large group at ThoughtWorks led by Harshal Hayat-Nagarkar, Chaya Yadav and others who work on Bharatsim. And for the Karnataka data, we've collaborated with Raji Sundaresan and Sivatreya at ISC Bangalore and ISI Bangalore. And there are many other people who I've learned very much from, who are not listed here, but they're all equally important in the development of my own understanding of this subject. So let me just move back to this particular slide and let me stop now. Thank you.
Thank you so much, Professor Menon. It was so engaging. I completely lost track of time, and it was indeed a day of learning for me, and I'm sure for all our listeners as well. Thank you so much. So I have a few questions from our listeners. I have a couple of my own, but let me start with the listeners first. So this is a very interesting question, actually, and it has been doing rounds since we started with this COVID-19. So this particular person has asked that it looks like some people keep on saying that the coronavirus is a man-made virus and it was purposely spread. And what do you think about it? What are your views? Because this is something which keeps on doing rounds every now and then, this kind of news.
So I think this particular point has been conclusively addressed in work that many people have done. There's really no reason at this point to think that it was man-made. Of course, there are many, many examples of spillover events in which viruses that have infected animals. For example, bats are a major reservoir of these viruses. And we know the procedure by which they move first to some intermediate animal and then to human beings. At this point, there's no reason at all to believe that some lab in Wuhan may have been responsible for releasing this either by accident or on purpose. And if it was on purpose, that would have raised the interesting question of why would they release it in Wuhan, if it was a Chinese-owned city. That there are certainly technical reasons not to think that this was completely natural. And the real reason for this is probably because of the increased contact between humans and wildlife, which is also a reflection, is an ecological problem that as we as the extent to which humans attempt to control their environment infringes on other types of species that are there, spillover events will become natural. So in a sense, we have to learn to live in peace and in coexistence
Yes,
with our natural environment. I think
that
that's really the important
that's
point.
a great thought. That's a great thought, Professor Menon. And I mean, another listener has asked this question actually, that increased surge in this, zoonotic infections that we see. Shall we refrain from keeping pets or something like that? I mean, does that sound like a solution to this problem? Like, or
So none of the diseases that, for example, the 3 out of 4 that I mentioned of emerging diseases really seem to be associated with pets. A lot of it seems to be bat related, so bats to intermediate animals to human beings, to poultry. So poultry and birds are another thing. And of course, you don't keep poultry as a pet usually. I think the situation more is that in situations where animals are factory farmed, if they're there at very high densities and large numbers, it becomes easy for viruses to circulate between them, to be modified, and to undergo the sort of modifications that are required for them to move to human beings. It's not that easy to do. It's not, many viruses will stay completely happily in their hosts without crossing over, without spilling over into human beings. So that's not an important, I would say that with pets, you should not worry at all. The emotional sustenance that you get from having a pet is probably worth far more than the risks of getting a virus from it.
yes, indeed. So there is one more question. That we have seen that reinfection is occurring in people, who have suffered from COVID-19 earlier, and now they suffered from COVID-19 again. So first of all, why is this happening? Like, I mean, I may understand that there is strain variability which is coming in now, and we see that probably it was difference in the strain, but intensity of the disease should be lesser the second time. That's the assumption that we will have, based on the strain matching and everything else. What are your views on that, sir?
I think we don't know enough of the biology and immunology involved in this particular case to be sure. We don't really know of the levels or the importance of reinfection. Is it just happening in 1% of the cases? Or is it something that sooner or later everyone will be reinfected because the virus happens to be circulating? We need better genomic surveillance to exactly address the question that you asked. When you're reinfected, have you been reinfected with a different strain? And how different did that strain have to be from the original strain so that your protection that derived from the original strain did not carry over to the second strain? So we need to learn much more on the clinical, the medical, and the immunological side before that question can be answered. Right now, we do know that reinfections are a possibility, but we don't know enough about the scale of such reinfections and indeed how long does protection last, either from having been infected earlier or from the vaccine that you will be getting in the future.
Um, Sir, another question is relative to past pandemic, the rapid access to information and data in this pandemic has influenced what aspects of how we deal with the pandemic the most, as per your opinion?
Oh, that's a great question. I think the ability to understand, to sort of know what other people are doing and to sort of participate in this global scientific enterprise that is devoted to the understanding of COVID-19 has been absolutely crucial and central. The fact that as soon as a strain, a sequence that is sent to a common web server where anyone around the world can access it and look at, see how did that strain differ from earlier strains? How did that particular variant change? What is special about the South African variant? What is special about the UK variant? All of this is very, very crucial to our understanding of the disease. A certain clinical approach that has been tried out in a hospital in Italy or a hospital in China is immediately available. People are writing preprints that are available to everyone on the medRxiv or the bioRxiv. So this access to this global fund of knowledge is completely unprecedented. It's never happened before in our history. And in a sense, that's an extreme positive of the COVID-19. If ever there's a positive to anything like this, it's the fact that scientists can collaborate across countries, across continents, share information. And there is no sense in which, look, I want to protect what I know, I don't want to tell you. That has vanished altogether. And I think this is the great positive.
Has skepticism also grown because of that? Why I'm asking this, because, the recent controversies regarding vaccines, for example, listener has asked us, can you tell us something about the controversy regarding Covaxin and they released the vaccine without completion of their phase 3 trials and without publishing their results openly. So do you think that this kind of skepticism is also increased because, it's there, the information is out there and everyone knows?
I agree. So there are two sides to this. One side to having information available at your fingertips all the time is that some of it is good information, a lot of it is bad information. And as a consumer of information, you have to make your own decisions about what is right, what is not. You have to consult other people, and you have to look for trusted sources of information. What can you trust? What can you not trust? So that's one question about information, who you trust. You have to make that decision yourself. And as people who talk about science and who popularize science, it's our responsibility to tell people, trust this, don't trust that. This works, this is the reason why you should trust this, and this is the reason why you should not trust it. Finally, everyone has to make, you can't order people to believe something. It only works if they understand what it is and make that decision on their own in a conscious way. So that was a separate answer from the vaccine answer. And regarding vaccines, public health, to the extent that I understand a little bit about it now, it's a tricky thing. Finally, there are questions of policy, there are questions of politics, and someone has to take hard decisions based on incomplete information. The fact, information will never be complete. And you will always be in a position to have to take a decision, especially in a very rapidly moving scenario where people are falling ill, there are new variants appearing, that may not be supported apparently by what, no, you have to make an intelligent guess for what might happen in the future. In general, however, I should also point out that transparency is vital to the scientific enterprise. It does not matter who is doing it. Is something an Indian company, something a foreign company. Every scientist has the responsibility to be transparent. Every consumer has a right to demand transparency, especially in matters of public health. When you accept a vaccination, you're a healthy person being vaccinated as a disease. It's not as though you're already being diseased and you're looking desperately for a treatment that might help you to recover. So whenever you administer anything to a healthy person, whether a medicine, whether a vaccine, your standards should be high enough. You have to couple that, the desire for transparency and being explicit about the information that you have, with the requirement to make a decision. So only these two things have to go together. You must both respect the need for making decisions with incomplete information, as well as the requirement that you must be as transparent as possible, absolutely to the limit that you can be transparent. And you must be able to explain your decisions. These are all the issues that you must keep in mind when thinking about this question. I'm not sort of committing either to plus or minus, you should or you shouldn't. I'm just saying this is the information that you should have, and you should ask, have I received all the information that I need to make the decision? If I have not, why have I not received that information?
That's a great thought, sir. That's a great thought indeed. I had a question actually. In the very beginning, we had another noted speaker who had spoken about R naught and everything else. I had asked few questions regarding R naught then also, and I'm going to be bothering you again with those questions. So my understanding about R naught is that to calculate effective R naught, you should have the transmissibility values and the contact rate and duration of infection and everything else. But we see a variability, isn't it? Like in the start, in the beginning of this pandemic, we didn't have enough numbers, so probably we saw variable R naught values, right? But within the states also we see this difference. So what is the prime reason and how can this be circumvented?
That's a great question. So let me just make a distinction between different values. R naught is usually calculated at the beginning of the pandemic, when the epidemic is just taking off in numbers and people have not put in any sort of restrictions associated with the disease. So that's as though I'm not interfering with it at all. I'm just seeing the numbers rise exponentially in the beginning. So that is the basic reproductive ratio. As you move further on in the pandemic, people begin to change their behavior. They tend to avoid people who might be coughing loudly. The restaurants will only admit 50% of the people. People will self-quarantine. And then it becomes much more difficult for the disease to go between person and person. So then you have to talk about an effective reproductive ratio, not a basic reproductive ratio. That's one thing. The other thing is that the quantity itself, the reproductive ratio itself, is determined by multiple things. It's determined partly by biology and partly by social interactions. The biology part of it is, where is the virus? Is it in your upper respiratory tract or your lower respiratory tract? How much of it is multiplying? What is your viral load? How much are you coughing, etc., etc.? That determines how easy it is for the virus to go from one person to another person. But it's also dependent upon contacts, as I said. If you have various interventions that ensure lockdowns, that ensure that people don't come into contact with each other, you switch off public transport, you just make it that much more difficult for people to contact each other. Once that is done, your reproductive ratio will go down. So the combination of biology and the combination of social interaction is together a very hard thing to model, to understand together. But that might account for the fact that different states have had different states have different densities, different states have different ways in which people interact, different states have different age profiles. And that can make a considerable difference in terms of people getting infected and being able to transfer the infection to other people. And all of this is, it's a complicated question. There is no single wholly reproductive ratio number. One has to sort of look at it in its context and account for that variation across India, exactly as you suggested.
Sure, sir. So, sir, I mean, another thing that people, I mean, there are some basic questions that we have received. I'm not really sure if we have time for that, but I'll take this one up probably. That could you explain about beta gamma ratio in the basic reproduction model equation, or you could direct them to a source where they could get this information from.
Yeah, much of this is sort of standard information about, I can send you a reference at the end of this, which you can circulate to anyone who wanted to ask. And the beta over gamma ratio, which I defined as the basic reproductive ratio R naught, really encodes exactly what I discussed just now. It's that duration of your infectiveness. That is the 1 over gamma. How long are you infected? And the other part is how effective are you at transferring the infection from one person to the other person. That's where the beta comes in. So the beta by gamma really is exactly a measure of what I told you about. The efficiency of transferring the infection to another person, the average number of people you meet, and the duration over which you remain infectious. So all of these together will determine, on average, how many people will you transfer the infection to. So you can go back to those equations and actually verify that the same idea that I described is actually hidden inside those equations. And I'll send you a reference for this as well.
That'll be really helpful for all the listeners, sir. So how can students contribute towards data analysis using IndiCISM that you just mentioned? And are there any internships available? Are there any career options available for students who want to participate in this?
So I think student involvement in the Bharatsim project, I think, is something that could potentially be very important, because we'd like to be able to crowdsource as much data as we can. The whole idea behind the Bharatsim project is to have an as accurate slice of the Indian population as we possibly can, to the extent that we need to understand what is the age distribution, what is the levels of education, health status, et cetera, and put it into this thing that later will become a public resource that anyone can use, anyone across India can use. To do this, one needs data, and one needs to be able to put the data together. And so we will see if we can advertise internships at some point that have to deal with this, so that people who will just help us assimilate this data from multiple sources and put it together in a form that the Bharat Sim program can take in.
So a platform like this is being created as we speak, or is it available right now?
The development of Bharatsim is going on at the moment. Hopefully within another month or two, we'll be able to demo it in some public way. And over a longer term, there will be continuous development of Bharatsim. And that's the point at which we would hope that people would be able to come in to contribute towards it as a public good. This is going to be a publicly available resource for anyone in India to be able to use to discuss questions of public health.
That's really great, sir. I mean, I am sure that government and all of you are doing more than enough for the younger generation so that they can participate in such things and opening avenues for them career-wise and otherwise also. So, I mean, I have some more questions, but I guess that we have run out of time, and I'll be happy to share these questions with you and see if we can answer some of them.
Happy to answer them.
I again would, from all of us, on behalf of every one of us, I would like to thank you for such detailed and interesting talk that you have delivered today. And like I said, that I personally have learned a lot, and I'm sure, I mean, others listeners have also done so. So thank you so much, sir.
Thank you to the audience for their patience. Goodbye.
Thank you, sir. I have a couple of announcements to make regarding the feedback form. So as usual, all the attendees are requested to submit their feedback form, which the team will be sending over to you. Please make sure that you provide your input, following which you will receive your certificate of participation. For those who have joined through the Manav Facebook page, they can send their email IDs into our Facebook Messenger box, and we'll share the link for the feedback form, and you'll receive your digital certificate after you send in the feedback form. Regarding our next webinar, please stay connected with us through our social media handles on Facebook and Twitter. And I would like to thank all our listeners and again, Dr. Menon. So from Mano Human Atlas Initiative, thank you so much.